Alright, this next video is going to take a look at the inner workings of the cell, looking at the cytoskeleton, the structural components that make up the cell. Now, ultimately what we're looking at are two different structures. Those different structures we're talking about are microfilaments and microtubules. What their function is for is structural support. They're going to help maintain the three-dimensional shape that's going to be within inside the cell and provide as an anchorage or storage area where organelles, not necessarily storage, but anchorage for organelles to attach onto and move on. As you guys watched in one of the videos, the cell motility allows for materials to move. The motor proteins would walk along on the microtubules, so allowing for organelle locomotion as well, like the vesicle. The cilia and flagella can move in a 9x2 orientation, which we'll see here in a second, of the microtubules, and it can allow for regulation, organizing structures, and cell activities. So in terms of what they are, the ones that I really want you to focus in are the microtubules and microfilaments. Those are the ones that are going to make up the major component. Intermediate filaments are going to be things like actin and myosin, things that we're going to see within other structures of cells. But microtubules and microfilaments are the main ones that we want to know. Knowing what they are, if I have a normal cell, one of the biggest things we could only see would be a nucleus. But now if I can see a three-dimensional shape, I can see these structures that are within it, like these big long blue tubes. These blue tubes are the microtubules. And then I could also have some microfilaments in there that could also be a transport system or a structural component of the cell and cell membrane. Now, the perspective of it, proteins that make up fibers are very similar in all living things, whether it is in bacteria, whether it's in protists, in fungus, animals, plants. They all come from the same particular components. Now, if you remember back to one of the videos we watched as a sophomore, that you are 50% identical to that of a banana. So there's a lot of the same components in terms of your genetic material that makes up this stuff that is going to be within you as well as in a banana. Now, bananas are also going to be able to have cell structures and the production of some of these proteins. So do we. For example, in us, all eukaryotic cells have actin, one of those protein filaments. Okay, microtubules, these are the largest of the ones. This is one to make sure you know. If anything you see on a test where it says alpha tubulin, beta tubulin, that's going to be the components or the subunits of it, you could probably have an assumption that if it contains tubulin and tubulin, it's probably a tubule. Okay, these are the thickest of the fibers, hollow rods. They construct proteins, and this is the one where you saw the motor proteins move on. Okay, showing the structure, they move, structural support and movement. They also are going to be centering off from the centrioles. Track and guide motor proteins, carrying organelles. This is another example where carrying along that vesicle. Okay. Example shown here in a 9x3 orientation. So if I count up, I got 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them, but with 3 of them each. There's our 9x3 orientation of our centriole. And this is going to be utilized as a job for cell division. Whereas cilia and flagella have a 9x2 orientation of those microtubules. Cilia are numerous hair-like projections, where flagella are long whip-like tails. Okay, they create some sort of an oar-type movement as if move back and forth, creating locomotion. Where undulated movement is going to be created with the flagellum. Here showing you an illustration of a 9x2. So 2 microtubules in connection, but there's 9 pairs of them. So example, bending of the cilia and flagella is driven by a motor protein, and that motor protein here is dynin. So that's the structure that you saw moving. And it's going to require ATP. So if it requires ATP, there also must be an organelle around nearby, which is going to be your mitochondria, to allow for energy production. Okay. Last ones that we're going to talk about are microfilaments, like actin filaments. These structures are the thinnest class of fibers. Solid rods of protein. You have actin within there. We see a lot of those in muscle cells. Function in three-dimensional work. I'm just going to move out of this quick. Let's see. Slow this one down. Actually, we can get rid of that altogether. That's not something we're going to need to look at. We'll look at the structure here. Go back. Functions in three-dimensional network inside cell membranes. In muscle cells where you have actin and myosin filaments. These are the little protein filaments that if you've had anatomy physiology, you understood that actin is going to slide together with myosin when calcium is present and causing a muscle contraction. These are individual little filaments that are going to slide together, and as they slide together, they allow the muscle to shrink. So here's an illustration showing that. So that dark purple is the myosin, this gold coloration is the actin. As they slide together, it shrinks this little space called the H zone, which causes a contraction to take place. Okay. Intermediate filaments, we're going to skip past this. Nothing that we're going to see on your test. It's just, again, another structure that's going to help maintain your cytoskeleton. It's going to reinforce the cell shape. So ultimately, the two things you really want to make sure you look at are these two microfilaments and microtubules. If something is made up of tubes or alpha or beta tubulin, it must be a tubule. They're the thickest. They allow for cell structure and cell movement materials inside and on the side of the cell. Where filaments are much thinner, this is more internal movements within the cell, like actin and myosin, the contraction of muscles. That's it for the type of cytoskeleton. Now, moving our way into how cells can interact and communicate with each other is known as cell junctions. Here, there are a few different illustrations of some of those junctions. Illustrations showing you how they can almost solder themselves together from one cell to another. Now, there are different ways that we can do this. The first one that we're going to talk about is going to be found inside a plant cell. Plant cells, since they have the really rigid structure of the cell wall, they need to be able to have one cell communicate with another, but they can't do that through the cell wall. So they actually have to have pores through those cell walls. And so the term you need to know is this term right down here. That's the plasmodesmata. The plasmodesma or plasmodesmata collectively are the pores that allow cells to communicate between each other. And showing the illustration here. The channels between allow the cytosol to pass. So what that means is not only cytoplasm, but other organelles can be able to interact and pass through. So it's a big, large opening for materials to pass around. Animal cell surface, so you have an extracellular matrix, so you have other fibers on the outside of the cell that can allow for increase in surface area. So I can attach onto things, adhesion, where I can attach onto other portions of other cells around the outside. I can allow for movement if it's cilia or flagella, helping to regulate materials or a structural component in support. In animal cells, there's a few other ones besides the plasmodesmata. This one here is called a gap junction. Gap junctions are just like, they are channels, and what they do in these gap junctions are just like plasmodesmata of a plant. However, they're between animal cells. It's just an open pore. The two other ones that we're looking at are tight junctions. Tight junctions are like a little soldering point, just a little glued together space between them. There's not any interaction between them, but what it allows it to do is just kind of solder together, stick together. No communication between the other. And the last one here is what's called a desmosome. And at this point, they have soldered together, but not only have they done it with just in between, but they've also put in anchors, almost like a root system. As they put in those root system, it allows it to really withstand and have a hard time pulling apart from each other. So they have tight junctions, which is more of just a glue point next to each other, a little soldering point. Gap junctions, which allow open communication for the cytoplasmic movement to be able to treat, just corresponding to that of the plasmodesmata in plants. And the desmosomes that have the anchors that allow them to fasten together. Now, ways in which they can signal, they can have direct contact signaling. Materials can pass information back and forth, so like information transfer. So pancreas can signal out materials, endocrine system, synaptic nerves can send information through neurotransmitters. A few other ways that our bodies can utilize, or cells can utilize some of these junctions to be able to send information out to the next one. Okay. At that point, that's where we're going to stop for here. Or the rest of the stuff we are not going to worry about for your test. All right.